Okay, I guess it's time to begin. Welcome to all of you to this at-home edition of the Slac Public Lectures. We hope that everyone who's attending tonight has seen the video for which the link has been posted. In just a minute, we'll have a very brief 1-minute recap. And then if you've come tonight, probably you have questions that you'd like to ask our distinguished set of panelists. So let me just give a brief recap of, oh, okay. Very good. Okay, hello. My name is Michael Peskin, and I welcome you to the current edition of the Slac Public Lectures. Things have changed a little since we were in a room with 300 people all attentively listening, but I hope tonight will be interesting. The speaker will be Nick Edwards, who's a research associate here at Slac, and we have actually a distinguished panel of panelists who will accompany Nick and describe some of the research that he's talking about. So once again, if you're here, I hope there's Nick. I hope you know that Nick is a paleontologist, educated at the University of Manchester in the UK. During his PhD, he became an aficionado of these big machines that we have here and discovered very interesting applications of those to paleontology, which were described in the 1-hour video that we asked you to watch. There are also 3 of his colleagues here whom Nick will introduce after the video. And so maybe without further ado, I should turn it over to Nick to just review the topic of this session, and then we'll begin the discussion. So Nick, you want to share your screen?
Yeah, sure. Thank you, Michael. Hello, everyone. Thanks for those here that are attending. Let me just bring up a quick intro to the Q&A, a little refresher for people if they watch the video, and if not, here's a quick outline of what we did. So what I talked about in my public lecture was how my colleagues and I, some of which are here tonight, use a synchrotron to study fossils. And a synchrotron is a particle accelerator. In particular, we are here at Slac, and we use the Stanford synchrotron radiation light source at Slac. Now, this is a particle accelerator that generates extremely bright X-rays. And here is sort of inside the particle accelerator, so really big, cool physics engineering. As I said, we use the X-rays that the synchrotron generates as part of the electromagnetic spectrum. And we use X-rays because we want to do something called X-ray fluorescence imaging, a particular way X-rays interact with our samples that allow us to detect and image the chemistry of different materials, in this case, fossils. It can also do some really cool stuff with X-ray absorption, where we can detect how different elements are bound to each other. And then I went and talked about a few case studies, including Archaeopteryx, where we were able to see previously hidden chemistry in its preserved feathers from 150 million years ago, a early bird, and where we found the elements associated with pigmentation, particularly melanin, in the preserved feathers of this bird. We talked a little bit about the different types of melanins and how we went and looked at modern birds as well to try and discover more about that chemistry, and then how we could use that information to look at pigments in a 3 million year old fossil mouse. So that's it, the really brief overview there. So hopefully everybody could see and hear that.
Thanks again, Nick. Before I turn it over to the panel, let me just describe how this session will proceed. if you look at your Zoom screen, down at the bottom toward the right, there's a little button called Q&A. If you press that, a window will open. If you would type in your question, then we will see it, and in order those questions will get asked to the panel. If you want to put some identifying name, like Tyrannosaurus Lover from Redwood City or whatever, however you'd like to be known, we'll announce that too. And maybe I should say this whole session will be recorded. So if you have interesting questions, people will be able to hear them when it's over. So maybe, I don't see any questions there yet, but hopefully people will type some in. And in the meantime, let me turn it over to you, Nick, to introduce the panel.
Thanks again, Michael. So yeah, tonight, hopefully they'll start coming online with their video feeds now, but we have first we have Jennifer Anne. She's come on first, so she gets to be announced first. Jen, aka Indy, is currently a paleontologist or lead paleontologist at the Children's Museum of Indianapolis. But before that, she spent a lot of time and did her PhD at Manchester, which is how we met, and she got into that. And a lot of her research during her PhD was focused on using the synchrotron, in particularly to study bones and bone chemistry in the same way that I focused on looking at feather and color chemistry. She was interested in what different elements could be teased out of fossil bones. So that's Indy. We also have Uwe Bergman, who in my screen is on the bottom right there. Uwe is a physicist and distinguished scientist at Slac. He initially developed the rapid X-ray fluorescence imaging technique that we primarily use for our research many years ago really unfortunately. Doesn't seem like it was that long ago. But he's our physicist synchrotron guru and really introduced us to the world of using the synchrotron to study ancient artifacts. And then finally, we have Roy Regalius as well, who's a geochemist from the University of Manchester in the UK. Roy was mine and Indy's PhD supervisors and my boss in my postdoc as well. And he is our guru on how elements and different things react in the earth over millions of years. So, hello everyone. How are we all doing?
Okay.
I think Roy gets an extra shout out because it's 3:30 in the morning.
Yes, he should.
So.
Yeah, Roy is currently in the UK and it's 3:30 a.m. So, so thank you, Roy, for making the effort to join us here. So, I also just wanted to mention at this point to everyone that this, and I mentioned at the end of my lecture too, that really this research that we talked about in the lecture is only possible with the combined expertise and knowledge of people from multiple different disciplines, because we have these multiple disciplines combining together in order to understand the data. You know, we need a physicist to understand how to collect the data, a geochemist to understand what the elements mean, and a paleontologist to understand what they might mean from the paleontology sense. So I'm glad that we were able to have this mix of people in the panelist here. So.
Okay, so let me start you off with an easy question, and then the questions are now appearing on the Q&A.
Right.
So if people out there have questions, open up that little Q&A window. It's at the bottom on the right of your Zoom screen, and type in your name and your question. So the first question is, why do we want to know what color a dinosaur's skin or a bird's feather is? Now, there's an obvious answer, which is that when you went through school, everyone told you it was impossible, that that information had been irretrievably lost. And so there's an incredible coolness to finding out really scientifically how that works. But maybe there's a better answer that you would write on a grant proposal. I wonder what you folks would like to say.
Well, I'll start off by, I think we each have some input in here, because again, we all have a different perspective on this. But actually thinking about this and talking with Uwe a little bit earlier, was that there's, as you say, it's very cool inherently. There's something that's naturally attractive about this science, and dinosaurs. You know, there's a reason why we all get fascinated about it as kids. But I also felt that there's something about the idea of being able to bring the aspect of color into paleontology really kind of makes it more tangible for us, and almost brings it closer in the same way that we might see black and white films, you know, from even like, you know, the mid 20th century. Seeing some of those rendered into color somehow brings them closer to us. And I feel like that's a big aspect of why we're kind of interested in looking at the color of ancient organisms. So I was wondering if anybody else here, Uwe, do you have an outlook or perspective on that?
Well, yeah, thank you. And I have to say, from my background as a physicist, it would not have been like one of those questions which my colleagues would typically ask. However, through this research, and thanks to Roy and my colleagues who taught me a lot about that, including some evolutionary biology and also paleontology, I started to appreciate how actually critically important colors are in life. And it is not any color of any animal which is around us today is not coincidental. There is a reason for those colors, and by knowing them, we can then also learn about the behavior and about the animal itself. And I think that is a strong, it's a strong enough motivation to be curious about it. And the other aspect is that what I also learned from Roy, and I hope Roy can elaborate a little bit more on that, is maybe the origin of color wasn't originally the same reason as it evolved later on. So there might have been another reason to make the color of feathers or skin important, which then was used as an advantage when the survival of the species depended on adapting that color to the circumstances. So maybe I can hand it over to Roy here.
Yeah, sure. Thank you, Uwe. And for me, good morning to everybody. I'm talking to you from Manchester in the UK, so forgive me if I'm a little bit slow to respond. But it's a really fundamental question. I think it's a really good question. And there were, before I Uwe did a good job of introducing one aspect. But before I get to that, I just wanted to make the point that what Nick was talking about was being able to envision these organisms, and that's fundamentally exciting. But understanding color, and I should say that it's not just color, but it's also patterning. It gives us a little bit of an insight into the behavior of the animal and the type of environment it was in. If you think about a zebra, you know, if we understand that patterning on a zebra, or zebra, which I should say for this audience, you know that it's using that patternation as camouflage. So this not only teaches us about the animal itself, but it also can teach us something about its environment and the type of lifestyle, if you will. What Uwe was talking about is, I think, also fascinating. What he was saying is that the evolution of the chemicals that produce color very often have origins in other processes. And for me, that's been one of the most fascinating parts of this research. All of you, everybody that's participating in this webinar has pigment in their hair and in their skin. And I think it might really fascinate you to know that the origins of one of those fundamental pigments, which Nick talked about quite a bit in his lecture, eumelanin, the dark black pigment, which all four of us in the panel have fairly high amounts in our hair. that actually originated from a very, very different evolutionary process. That had to do with detoxifying the enzyme that allows eumelanin to form. That actually has its origins way, way back in the very, very early parts of life, in order to allow organisms to detoxify themselves from poisonous atmospheric oxygen. So the other side of being interested in being able to resolve patternation and color in organisms is to try and find the biochemical pathways that lead back into deep time.
Thanks, Roy. I think Indy, can you give us a brief answer, or do
Can
you?
I ask another question? To
Oh yeah, let's go on to another question.
Okay, so Annie asked the question, Jennifer, you create visual displays of extinct animals for your museum. And Nick just told us that we have incredibly little information about that. So how do you figure out what color to paint the dinosaurs and what color to make the various animals that you put on display?
That's a good question. And actually, I was going to answer the whole color thing from a public programming and education point of view, since Roy and Uwe did just a good job about the science part. So first off, it's one of the things that we can tell the kids we don't know. And that gets the kids really, really excited, because they realize that science is still happening. Like, there's still things that they're going to be able to do when they're scientists. And I think that's important, not just for the kids, but when you're talking about writing grants, showing that there is still so much we don't know and things that can be changing, especially as we get new technology. So that's one thing. But the other thing is that we tell them that we don't just pick a color. My favorite color is blue. I don't make all my dinosaurs blue. We use scientific evidence. So we're using research like what this group right here is doing, and we'll mention it in our exhibits. I try to keep all our interpretation staff up to date in the latest research. But we also look at the living relatives of dinosaurs. So those are crocodiles and birds. Both use color, both can see in color. So it's pretty fair that dinosaurs did as well. So then when you're starting to do the color, we tell the kids, you know what? Sure, make your T-Rex purple, prove me wrong. There are birds that have purples, there are reptiles that have purples, so who's to say that T-Rex wasn't purple? So we actually use it more as yes, there's constantly more things that you can learn. We don't have all the answers, and that's actually what makes science really, really fun.
So Buddy Haas from San Carlos asks, is there a great demand for testing paleontological samples here at Slac or at other synchrotrons? Is this something that's caught on in the community, and do you have a large demand?
Yeah, and well, I mean, large demand in terms of relative to the amount of other research that's done. It's a relatively small amount. However, we do have multiple groups, research groups from different universities across the world who are interested in studying fossils in lots of different ways. Some come and use a synchrotron, and it sometimes doesn't pan out the way they do. Maybe the preservation wasn't great, but then other times they get good results. But we do have some regular research groups who come and use the synchrotron to study fossils. So there is enough demand for us to keep scheduling them time to do experiments.
Okay. And from all over the world?
From all over the world, yeah. So, and people have lots of good fossils that they want to look at. So,
we have more fossils than beam time.
Yeah. So,
Ginger asked about Tully Monsters. I don't know what those are. She asked, do you agree with the sort of lamprey?
So I think Roy and Indy might be good to take on this question, because they have direct experience with this first.
Take it away, Roy.
Well, thank you. Okay. So the Tully Monster, this is a Tully Monster. This is quite an iconic fossil. It's tremendously well known. People have studied it for a long, long time. There's a controversy about the Tully Monster, about its origins. And we've done some scanning of the Tully Monstrum. we haven't published any of that work because there have been, actually this leads into the previous question. There are other research groups that are working on this. And the fundamental question is whether this can be classified as a vertebrate or an invertebrate. And it's a real scientific controversy right now. I don't want to go into too much more detail on that. Indy, do you want to add anything?
Nope. Like you said, it's one of those fossils that's, you know, right now it's a vertebrate. 6 months from now, it'll be an invertebrate because someone found new information. And because it's such an iconic and sexy fossil, this is something that you'll see a lot picked up in the news. So don't be surprised if you see something different, because a lot of people are doing work on it.
I think the main thing to point out is that it's a very early life form, and it could be one of the origins of all organisms with a backbone. So whether this thing actually has a backbone or not, you know, it could be the first fossil or animal that we know of with a backbone. So this is why it's very contentious.
From the Cambrian or before?
It will be indeed Precambrian, the kind of around that sort of time, Cambrian probably, yeah. So
very good. Dennis would like to know what's currently on the beamline. What are you looking at now with the X-ray fluorescence setup?
In terms of our research specifically?
Well,
yes.
You get things. What are you putting on the next thing? Yeah, I guess after
the next thing. Yeah. So I think the most current kind of research that we're doing right now actively is actually the work I'm doing with Roy. So looking at very early bone material and trying to understand how bone itself evolved. And do you want to elaborate on that a smidge, Roy?
Well, we don't want to give away any secrets just yet, but this is a big part of the answer to that previous question about why are we interested in this. I mean, we are trying to pull out the chemical details of the origins of different species, and one of the key ones is how did the backbone, how did we become vertebrates? How did that capability evolve? And there's chemical information buried in the fossils that this technology, which Uwe and colleagues at Slac and at other synchrotrons have helped develop, there's information there that no one has been able to see until now. It's really quite fascinating, actually.
Arjun has a question about the sex differences in colors. So we know, of course, birds are different colored if they're male or female. Have you seen such things or evidence for sex differentiation in color in fossils?
Indy would like to answer that question.
Please.
So first problem is not all birds do it. Some are the same. But our biggest problem right now with questions like that, and also with questions about reproduction tissue, is how do you sex a dinosaur? Ian Malcolm said it best. You just lift up their skirts. Like there's not a really good way to sex a dinosaur. So until we can actually say this is male, this is female, and we know it, unfortunately we can't even say anything about color, because even if you saw differences in color, is that male versus female? Is that juvenile versus adult? Because many juveniles have different pigmentations and different patterns than the adults. You can get regional differences. So for instance, on the East Coast of the United States, we have screech owls, and you have more gray pigmented ones in the north and more reddish pigment ones in the south. So there's all these different variables you have to think about. So unfortunately, until we can say boy, girl, even if we could see color differences, we wouldn't be able to tell you if those were from sexual dimorphism or if they were from any of those other cases.
Well, thank you very much. Kalpana would like to know whether dinosaurs had bioluminescence. Is there any evidence for that, or any need for dinosaurs to have bioluminescence?
I believe that, and Indy might be able to clarify on that, is that bioluminescence is a rather unique process, and what bioluminescence is normally used for in terms of functionality. It's particularly as far as I'm aware, it's actually it's an organic reaction where it releases energy in the form of light. So I don't think there's been any specific evidence of bioluminescence right now, but there is equal amounts of evidence out there, or lack of evidence out there, to say that they, didn't. So it's quite possible they might have bioluminesced. I mean, we can't, as good scientists, we can't say one or the other without data to back us up.
Mm.
Yes, so bioluminescence is usually by luciferin, which is a particular molecule. And usually you see it again in things like DC fishes, but you also see it in fireflies. So maybe usually you see it in things that are dealing with a lot of darkness. And we do have dinosaurs that would have lived at the poles. I don't know why they would have used bioluminescence, but hey, there was something out not too long ago about dinosaurs that fluoresce. And actually we know that quite a lot of animals see in the UV light spectrum. So for instance, chameleons, they can actually shine little bits in their skin and it looks very cool. So maybe dinosaurs did as well. And again, that's kind of a guess and a cool thought, because again, a lot of animals do see in that UV spectrum. So maybe dinosaurs are communicating with one another or something using that kind of fluorescent spectrum, but there's no hardcore evidence for it yet.
Is there a reason why chameleons fluoresce?
Don't know. A lot of it they think is from communication, because a lot of things that see in the UV spectrum that are predators, they're following things like urine trails from their prey. Bees use it to somehow that's helping them with the flowers, so they see flowers in a completely different way than we do. So there's communication again with fluorescence. So some birds, for instance, they think are actually seeing, if a male's displaying, the female can actually see more than what you're seeing in that display dance. So there's a lot of reasons to use kind of that fluorescent spectrum, but what dinosaurs in particular would use it for, don't know.
Okay.
All the time we just say life is weird.
Nature's weird.
Okay. Ralph has a somewhat technical question, so let me read it out.
Mm-hmm.
Another recent method for determining the colors of fossil organisms utilizes a scanning electron microscope analysis of melasomes, which I guess are particles in the cell that contain the melanin or some other pigment.
Mm-hmm. Yep.
So you can compare the shapes of those structures in living and fossil organisms. Presumably you know about this. Have the results of those studies been compared to your results? And do you agree or disagree? Well, take it away, please.
Roy, seeing as you made the effort to get up at 3:30, I'll let you answer this question.
That is a great question. It's a really
very excellent informed question. So
sorry, Nick.
It's an excellent informed question.
Yeah, it is. It's really good. That's absolutely right. There's a second way of trying to determine the presence of particularly these two pigments, these two dominant pigments, eumelanin and pheomelanin. And the melanosomes are the little organelles that have been identified as being present in high concentrations if you have a eumelanin or pheomelanin rich feathers or skin. It's pretty simple. The eumelanin melanosomes are kind of long, like almost like shaped like E. coli bacteria. And the pheomelanin melanosomes are sort of circular. But of course, part of my answer lies in what I just said about them looking like E. coli. If in a very, very well constrained measurement, then yes, if you know there's been minimal alteration, then you can use this structural information to make conclusions about pigmentation. The problem is that bacterial mats can often look exactly the same. And it becomes very, very difficult to be 100% sure that what you're looking at represents melanosomes as opposed to some kind of bacterial mat. And in fact, until quite recently, people thought all of this structure had to do with microbiological growth and fossilization of microbial mats. This is part of the reason why we developed this whole line of inquiry, because the chemistry is not mimicked in the same way by microbiology. And so the chemistry is a very, very useful independent test on whether we have pigmentation residue or whether there's some kind of overprint. And the other thing to say, remember, a lot of these things that we're looking at are 100 million years old, and very often the structural information has been wiped away, whereas the metals, which still have a bit of organic configuration, may persist over much, much longer periods of time, as long as a 500 million years, longer than that, in fact.
I'll add to that too, that, I mean, people studying melanosomes really are what kind of kick-started the pigmentation in paleontology research. And it was people looking at exceptionally preserved things like feathers in a scanning electron microscope and finding these bodies. Now, the community has kind of gone back and forth a little bit on whether these are the ambiguity of these, whether they are bacteria, are they actually melanosomes, back and forth like that. I would add that I didn't get a chance to talk about that in detail in the lecture because I had so much to talk about, but we have actually collected SEM images from bits we were allowed to sample of the fossils and have actually found melanosome-like structures associated with our chemistry. And I think before just to finalize, before we move on to the next question, this is why it's also very important to try and do a multi-technique analysis of any material you're looking at. Using one technique only gives you one view of your sample. And it's like a puzzle piece. Each technique gives you a different puzzle piece looking at it. It's like individual people looking at different parts of an elephant. They'll identify an elephant in a different way if they only look at the tail or the trunk. You have to look at the whole thing in different ways in order to understand things properly.
Could I add just one other thing before we go on to the next one, if you don't mind?
Sure.
Is that all right?
I think
the SEM, this is also very, very important. The other thing about the SEM, of course, is that it analyzes very, very small regions. Mm-hmm. So something like about the size of the tip of that pen, whereas the rapid scanning that we do, we're able to do things that are half the size of me. And so going back to my example of the zebra, if you analyze the square millimeter on a zebra, well, you got a 50-50 chance of finding out whether it was white or black, but you'd have no idea about the whole organism. Whereas this rapid scanning, which looks at the whole organism in many instances, or at least a large part of it, can give us an idea about patternation as opposed to just identifying whether color is present or not.
Okay. Kalpana has another question, which is the following. So in your lecture, Nick, you said that feathers define birds. Mm-hmm. But I guess Kalpana has heard that also raptors, dinosaur type raptors, can have feathers, and the feathers may not be a flying function. It may just have assisted them in some other way. What's the actual evidence about that? Are feathers uniquely for flying, or are there other applications of them?
Indy is making lots of facial expressions. So
I would say you'd have to say no, because we have birds today with feathers that don't fly. So penguins actually have feathers. They're very weird looking, but they're feathers. All ratites, which are the flightless birds, so ostriches, emus, they have feathers, but they're not flighted. Some of them are extremely odd. So like a cassowary, the weird flightless bird from Australia, their flight feathers, it's just the rachis, so just the middle part. It looks like a bunch of pencils coming out of it. So feathers do a whole bunch of things. And the current hypothesis is that feathers did not evolve first for flight. That came later. Because a lot of evidence that we have for feathers in the fossil record are not what you'd call flight feathers. So they're probably doing things like keeping them warm.
Excuse my ignorance, but all the examples you gave of birds, which are flightless, evolved from flighted birds that lost their ability to fly?
That's the current hypothesis. In fact, they think that ratites, the group that saw the flightless birds, instead of being from one ancestor, they had a whole bunch that decided that flying wasn't for them. So again, as we said before, nature is weird.
Yeah. But you said now it's the other way around also, that feathers evolve for a different reason. So maybe could you expand on that a little more?
Yeah, so feathers do a lot more than just flight. They keep you warm, which is probably why they first did evolve, is to keep you warm, which is also good evidence for dinosaurs being warm-blooded, if anybody's interested, because no point in keeping the heat in if you're not producing heat. They're also used for display, as we talked about with males and females. They're used for species recognition. Obviously, they can be modified. If you're something like a penguin or ducks, they can repel water, so they can be helpful for swimming or just in weather in general. They can help keep cool, so ostriches actually use them to shield their offspring in the heat. They'll actually provide shade for them. There's some birds of prey that actually take turns on who's on top of who to shade them in the desert. So there's a lot of different uses for feathers. And as far as things like the raptors, there's a wide range. So for instance, Velociraptor, the only way we know they have feathers is on their ulna, so this bone right here. There's what's called quill nodes. Those are these little bumps on the bone. They're actually where the feathers anchor in. Where other raptors, we actually have the feather impression. So we can actually look at what they were talking about a little bit in that question about different shapes of feathers and sizes of feathers and types of feathers. So we're able to dive in a little bit more into that. And the other interesting thing is dinosaurs are birds, birds are dinosaurs. There's no separation. So it's a very mean question to ask children because of the same thing. So just remember, pretty much almost anything you could say about what makes a bird, at least the theropod dinosaurs, the group of meat-eating dinosaurs, they have the same stuff. Wishbones, hollow bones, feathers, you name it, they've got it as well.
Very good.
May I just add one very short comment? For the person who asked the question, I think it's also interesting to consider the fact that flight has evolved in 3 completely independent ways. So feathered flight through birds, then skinned flight through the old pterodactyls and also bats, etc., and then insect flight, which is totally different altogether. So equally as one cannot associate directly a feather with flight, one can also not associate flight with feathers. Mm-hmm. So I just want to add that as a food for thought.
Okay, let's move on to the next question. So on the chat, there's the following interesting question. In Nick's talk, he emphasized that you can see the metals, but it's much harder to see the organic structure, the carbon-oxygen structure, sometimes sulfur also, that wraps the metal atom. And the color depends on both of those things. So how do we know, once we found, for example, copper or zinc, that what we're seeing wrapped around it is the original stuff and what we're inferring is the original color?
I think this is another excellent Roy question. Because he did a lot of the work on the mouse bit at the end, which was exactly this, which is the absorption spectroscopy and how we use that to identify what elements are bound to different elements and what that might need and why we think they're from the original animal and not from the fossil.
Right, but there's also the possibility that it degrades. So
quite possible, yeah, and Roy will, I'm sure, touch on that.
Okay.
Yeah, you know, again, that's a really interesting question. It's fundamental to the research that we do. So good, you know, great to ask that, and I'm really pleased to have a chance to answer that. You know, and I don't know whether I should speak quickly because I feel like I'm taking up an awful lot of time, but the fundamental thing is that we get these images, and the images give us information about where the different elements are located. And that's really important because then you can tell, does this chemical, does it exist within the skin? Does it exist within the feathers? Is it concentrated in the eyes, etc.? And that gives us a clue to functionality. Because as Nick said, we also have to worry about whether the fact that some of these things have been added posthumously. You know, like I said, some of these things are millions of years old, and there can be all kinds of geochemical processes. So a big part of what we've done, and this gets really technical, and so so forgive me. This is why undergraduates are probably scared of me, because I get into this whole spectroscopy side of things. But it's the configuration of the organic complex around the metal that lets us have confidence that what we're seeing is original. And we go through a tremendous amount of effort to analyze the chemical configuration and then compare it with modern analogs. And I have to say this, Nick was a really great PhD student and postdoc, and he was like if I may, he was a bit like a dog with a bone on this problem, because a lot of what we found out is that what we think we know about pigmentation in modern organisms is not well developed. And so Nick spent a huge amount of time looking at the configuration of metals in modern organisms. So we then had a blueprint we could compare to the fossil record. And in some cases, the chemical fidelity over long periods of preservation is really quite remarkable.
Okay. Ralph has another aspect to this question that he wanted to ask you. So in Nick's talk, Nick emphasized that the pigments with metals you can identify with this method, but there are other pigments that contain no metals that are purely organic. So is there a hope for those? Or is it just really that there's one part of this problem you can solve and another problem that some other brilliant person has to come around and find out how to solve it?
I'll give Roy a break for a minute, because I've thrown quite a few of the good questions at him. So I'll let him recover a bit. Yes, that's absolutely correct. You know, each technique is usually quite specific at what it can tell us. And it just so happens that the synchrotron, based on the X-ray fluorescence technique, is, you know, going to tell us about and help us identify elements and image those elements. Now, obviously, that doesn't help us when you have something you're looking for which doesn't bind to a metal, which the synchrotron is good at finding. So yes, there's these organic pigments which don't have a metal component to them, particularly something like carotenoids, right, which are hugely utilized in life. You know, parrots, the bright, intensely bright blues, yellows, greens, pinks in flamingos are carotenoids.
We're never going to find out whether dinosaurs were pink.
Well, yeah, exactly. That's the thing. That would be wonderful, wouldn't it? I was going to put a picture of Barney on my lecture at some point, but I wasn't sure about the copyright infringement that would entail. But you know, the thing is, it's quite possible that reconstruction we have of that early bird that we keep in monochrome, like black and white, we made that specifically black and white for a reason, because we had no evidence of other types of colors. All we could say was, in the bits where we saw copper, we believe that indicated the dark hue melanin pigment, which would dominate. Where there was less copper, we think there'd be less dark hue melanin pigment, which meant it was probably on the lighter shade. Now, that doesn't mean that that white area wasn't pigmented by something like a carotenoid and could have been green, for example, but we can't detect it using the synchrotron data that we collected for that paper. Now, there's other techniques which can detect things like carotenoids in modern tissues, but the problem is, because a lot of these molecules are generic, I would say have lots of generic, what are called monomers, basic building blocks of carbon rings all attached together in different ways. As they degrade over millions of years, they start to just look like individual building blocks, like Lego pieces, right? You know, you got all these Lego pieces and you can build them in any way to make a different molecule. And so that's the problem over hundreds of millions of years, is that you see these individual building blocks, but you don't know how they go together anymore. Which is why these metals was such a key thing to kind of look for in the synchrotron, because it's very specific. If those elements like copper are in feathers, it's very likely it's because of melanin, because there's no other reason for copper to be there unless it came from outside.
Okay. Ray would like you to expand a bit on the cultural heritage research at Slac.
Sure. Hey,
So
a question for
everything's go on your beamline than dinosaurs and birds.
Yeah.
What else are you putting there?
Yeah, well, I would say that most of the time, a lot of the X-ray fluorescence is used for the different research areas that the DOE for DOE mission, right? You know, loads of people use X-ray fluorescence. It's a very versatile technique. So lots of different samples in there: battery tech, biomedical science. People do a lot of things. But yeah, the cultural heritage part is one cool bit. And I don't know if Uwe is still there. He seems to have disappeared off of my video feed, but this would be a perfect time for him to jump in and chat with us if he can.
I think he's gone, but maybe you can answer the question a little.
Okay, I'll answer the question. So yeah, the cultural heritage stuff. So actually, Uwe first developed this technique for doing the chemical imaging of large objects, specifically for looking at an ancient manuscript. And I mentioned that briefly and showed it at the beginning of the talk, called the Archimedes palimpsest. Now, a palimpsest is a document that's been reused, essentially. So parchment was at a premium in ancient times. We're talking about the early, you know, hey, he's back. Uwe, we have a question for you, talking about cultural heritage. So feel free to jump in at any point. But basically, some of this reused parchment that was reused for religious texts was actually overwritten on some quite early important texts themselves. In this case, ancient writings of Archimedes, some of Archimedes' theories.
Yes,
Well, it looks like we're having our first technical glitch today with Uwe, just when we needed him as well. So I'll carry on talking for him. So yeah, and so what we're interested in, or what Uwe was interested in, and what we've done on subsequent documents of this type, is trying to uncover those writings which have since been obscured by later writings. And the synchrotron can help us with that, because we would hope that we can see the chemistry of the inks underneath the new stuff is different from the new stuff. So that image that I showed at the beginning, and so that way we can reveal the writings underneath by being able to image their chemistry and differentiate it from the chemistry that's over the top. We've also done some other cool things. So we can also, I'm also going to share my screen here and go here. Let's get rid of that. So we also did something on an opera score as well. So I'm hoping that you guys can see this.
Sorry, it's just a white box.
Okay, so I'll ditch that. That's probably one of my dual monitor issues here. So I'll just stop the share right now.
Just tell us what should have been there.
Yeah, sorry, Michael. I was having some technical issues myself here. So yeah, basically what we had was an opera score written by a chap called Cherubini, and his final aria, apparently people didn't like it when they heard it the first time. And so what they ended up doing was actually, the composer, in such a rage, apparently wiped it out, his final aria. And so actually what they were able to do was actually image this ancient opera score and bring the music out from underneath that hadn't been seen for a couple of 100 years. And so that's another cultural aspect that we've looked at before. So a lot of ancient manuscripts and trying to tease out previously lost information.
Okay, very good. Maybe I should tell the audience we're coming to the end of the question period. So we'll just take a couple more questions. If you have one, please, again, go to the Q&A box at the bottom on the right, type it in, and hopefully you can get it in time. Again, from the chat, there's a question of are you able to look at fossils evolving over time? Or are these really, you get one snapshot and then by the time you would get another, the creature is extinct?
Indeed, you want to take that one?
I'm kind of confused on the question.
It's basically the resolution of evolution, right? How are we able to really trace feathers through
The
time?
literal question is, are you looking at more recent fossils to get stability data and change over time data?
I'm the black sheep in the group and I don't do color, I do bone. So I've looked at a wide range of ages for things, and we actually did do some stuff that, as far as I'm concerned, is high altitude, so it's very young. But we looked at some archaeological specimens of bone, and we were looking at collagen preservation, and so we did a time-step series of that, looking at collagen preservation. So that was kind of the evolution of preservation, if you will. For the evolution of a whole organism, that's kind of these big picture projects that we kind of wink hinted at with the bone stuff, but as you can guess, big questions like that, we need a lot of specimens, so these are projects that take a long time. But they are big questions that we're trying to answer, and now that we've kind of worked out a lot of the kinks, Nick's done a great job really getting us some equipment to idiot-proof it for us so we can be really on top of this. Now we've really caught our groove. I think we're going to be answering a lot more of these really big picture questions. Because again, they're very deep time questions usually. They require a lot of specimens as far as the fossil record. And they also require a big thing, modern animals. If you can't say what's happening in the modern animal, you definitely can't say what's happening in the fossil critter. And when you start getting into these very deep time questions, sometimes the modern critter equivalent is a bit interesting. But all of us have had very fun experiences with these modern critters. But we're trying to get some of these deep time questions, and Roy's got some pretty cool things happening right now that, as a bone person, I'm super excited to see what happens.
Interesting.
So I think it's a good time because people showed interest in my cat in the video. So she's joined us for the Q&A here. So
there was indeed a question about whether you would analyze the color of your cat.
You know, we actually did, but my cat back in the UK, because I had a black cat, and we didn't use the data in the end. I can't remember why. I don't think it was, we didn't get good data out of it. Hello, kitty. Here she is. And I think the question was that if you basically a blind test, if you gave me a piece of fur that I didn't look like and I just looked at the data, would I be able to tell what color it is? Hopefully I would be able to take a pretty good guess to what color it would be based on some of those images that we saw on the modern feathers, because we think we've established a kind of, not markers, you know, you can't say definitively, but a combination of data indicators that allow us to kind of make a reasonable assumption based on data as opposed to just wildly stabbing in the dark.
Okay, and maybe here's the last question. So Mike wanted to know, are there any spin-offs from paleontology back into the study of manuscripts? For example, now that you know a lot about chemistry, maybe you know something about ink that would be interesting for people who are trying to date manuscripts. So is there an application like that? Or can you foresee one?
Well, actually, I think Roy's nodding too, so he'll probably have some input. But for me specifically, is that people use different inks in the past, right? And different elements, or a lot of the time they were based off of grinding up different rocks that were known to have different colors. So like azure blue is a copper mineral. And also you've got iron gall ink. It's kind of self-explanatory in the name, but people also used to use squid ink. And I think I mentioned in the talk that squid ink is actually used as a standard for studying melanin because it's super pure and it's you can able to dope it essentially with different metals. And yeah, so the idea of being able to see the chemistry of these inks or whatever material they use to do the write in the cultural heritage stuff could give an idea as to yeah, the elements you could see what different inks they used, and that's kind of slightly derived out of the work we've done with the paleontology, looking at modern and fossil pigments as well. So it's kind of a nice full circle there. And I think Roy might have something to add to that.
Yeah, I mean, one of the most common inks before modern times was something called iron gall ink. It was made by crushing up plant residue, and it was very, very high in iron. But depending on which plant and where the ink was manufactured, some of these inks are very high in arsenic, some are low in arsenic. So we can actually get some idea of different types of ink, when different things were written, et cetera, et cetera. So understanding the chemistry, using these methods to develop an understanding of fossils does definitely have an application in cultural heritage, being able to tease out different inks. And in fact, it gets even more beautiful because, as Nick alluded to, there's a whole range of paints that have been used over time by different civilizations and indeed by different artists. And there's a whole big area of this. We could do, in fact, there's a whole conference that's pretty much dedicated to this, and it's synchrotron radiation in art and architecture and archaeology. And there's a huge effort going into identifying different paints used by different artists, different schools, and the amount of chemical information and therefore information about different painting techniques and technologies is really quite remarkable.
I could, if you can see me, I could briefly just add one sentence. It started out, it started out with, let's say, cultural heritage, a document, and then it was realized that the method can be used for paleontology. You do paleontology, you start to do spectroscopy. There was also
Hey, sorry, Uwe, I think we're losing you.
Yeah. Maybe I should just say that there are, in the Slac public lecture series,
done with the cultural heritage, but every time you measure a new sample, a new specimens of fossil, or even a cultural, yeah, everything.
Okay, Uwe, I'm sorry, we're having trouble with your connection. So.
All right.
Maybe I should just add to this that on the Slac public lecture website, which you'll get a link to in a moment, there's a collection of our old videos. And there are several of them that have to do with the cultural heritage aspects of this program. In particular, one called Preserving the Vivid Yellows of Van Gogh and Matisse.
Mm-hmm.
So have a look for that.
And we can also put a number of resources and links to various things on the Slac webpage too, if people are interested.
Okay.
So
look, I'd like to thank those of you who came tonight. It's been a very interesting discussion. I hope you've had a good time and you've learned something from these people who were kind enough to help inform us about this question of color, paleontology, and synchrotrons. Let's just do a couple last things. We'd like to, because this is our first experiment with a remote public lecture, we're going to put a poll on the screen. And if you would just answer the poll, that would be very helpful to us in planning how this will evolve in the future. So there it is. So please vote, and that would be very useful to us. The second thing I'd like to say is that this session is being recorded, as you know, and the recording will be posted on the Slac public lecture website together with Nick's video. So you can go there and in a few days and see this, or other people who are curious can go there and basically relive the event. Also, if some of you have additional questions that you'd like to add, we'll probably make a transcript of this discussion. So if you would like to have additional questions answered, we can put those on the transcript, email them to the Slac communications group or the email address of the public lecture series. And we'll see what we can do about that. So when the poll goes away, there'll be a final slide giving you all the website information for the things that I've just described. So once again, let's thank the panelists. Well, I can clap for them.
Hey, I'll clap for it. Can we clap for each other? There we do. There we go.
And thanks to all of you for taking your evening to learn a little bit more about science.
Oh no, I'm hoping that people have found this useful. So,
Thank you.
Thanks, everybody.
Thanks.